Chapter 13, the older technique, okay? So first of all, the nucleic acid, okay? To identify and to visualize the DNA, it's very easy. So you need to run the DNA. You have to place the DNA in the agarose gel, and then perform the and then place the gel in the electric field. So all the DNA will migrate, okay, from the negative charge to the positive charge, and then we can separate the DNA molecule based on the size of the DNA, right? And each of these is the well. So you're going to load the DNA into the well and then perform the electrophoresis so that you can separate the DNA based on the size. And here you can see the dye. These are so-called tracking dye, because the gel, you can see this is UV light, right? So you incubate, or you add the ethylene bromide to the DNA, and then in the presence of UV light, you'll be able to visualize the DNA. If you don't use the ethylene bromide or the UV light, you don't see anything, all right? So basically, this tracking dye can help you to estimate the location of your DNA fragment size. Okay, so this is a typical DNA gel box. You place the gel here, and then you have negative charge, and then you apply the electricity from negative to positive, so DNA can migrate from this end to the other end, okay? So the gel electrophoresis is routinely used for detection and size analysis of the protein and nucleic acid, okay? And the reason why the DNA can migrate from negative charge to positive charge is because DNA, RNA, they are negative charged, right? The negative charge comes from the sugar phosphate backbone, right? And so that migrate from the negative to positive, and the migration speed, the smaller fragment can travel faster than the larger fragment. That's because the small DNA molecule can go through those agarose network faster, all right? They can penetrate through those pores and then go through the gel faster than the large fragment, okay? And so in the end, you will see the large fragment at the top of the gel, and small fragment will be at the bottom of the gel. And the DNA size estimation that we would skip because we don't do the estimation, but this graph here just tell you the distance travel and also the size of DNA. So the large DNA fragment will be here, and then they have poor resolution. Small DNA fragment will be here, will have the better resolution. If you look at this picture, then you will understand. So from here to here, the same distance, you can resolve only 150 base pair. But from here to here, if it's the same distance, it's 500 base pair. So that's why here you have a better resolution. And the least, as you go up of the gel, the resolution is poor. So that's the this graph means. And the different percentage of agarose also can provide a different resolution of the fragment. So if the percentage is low, that means the pore size is large. The pore size is large, then it's difficult to resolve the small DNA molecule. But it is useful to distinguish the large DNA molecule, all right? And then for a higher concentration of the agarose, so that means the pore size is small. Therefore, it is very good to resolve the good resolution for a small DNA fragment, okay? So the small percent, the low percentage, high percentage. And then to run the DNA, you need to have a buffer. So the buffer, like for example, TBE or TA, the trees borate EDTA or trees acetate EDTA, and then they can buffer the electrophoresis so that the pH will not change, and to maintain the certain pH so that you can have a stable DNA electrophoresis, the migration. And also the tracking dye, as I mentioned earlier, you have this bromophenol blue tracking dye and xylene cyanol. The xylene cyanol for 2% gel give you the indication of 800 base pair DNA location. And then for the bromophenol blue, that'll be the location of 100 base pair, right? Bromophenol blue. So like here, the bromophenol blue will be here, all right? And the xylene cyanol blue is here. This bromophenol blue in the bottom, xylene cyanol on the top. So when you perform the gel electrophoresis, because you cannot see anything, so you have to rely on these tracking dye, and then to estimate how far your fragment run on the gel, okay? Because if you don't monitor, then those DNA sample can run off the gel, all right? Run off the bottom of the gel, and then you will lose your DNA. And then I mentioned that the convenient way is to use the ethylene bromide, right? So that you can see the DNA fragment in the presence of the UV light. And then for DNA extraction, okay, DNA extraction. So DNA extraction, so basically you collect the cell, all right, you break the cell, and then to extract the DNA, so you have a different technique, approach to purify the DNA. So for example, you can use the organic method. So you use organic chemical like a phenylchloroform, or you can use the inorganic approach, all right? And so that means you precipitate the DNA, or you can have the solid phase that use the spin column to purify the DNA. And here is just the procedure that if you use the organic purification of DNA. So you mix the cell extract with the phenol, so that the phenol, like here, and then you mix it well, and then the phenol will precipitate the protein, all right? And leave the DNA RNA in this aqueous layer, and bottom is organic layer, and then you transfer this to a new tube, and then perform the chloroform extraction. So you mix this with chloroform, and then centrifuge the mixture so that the chloroform can remove all the residual phenol from this layer, all right? So that's how you purify the DNA through the organic approach. And then proteinase K, so in organic DNA isolation, basically it's a salting out, so you add a sodium chloride, and then at cold temperature in the presence of ethanol, then you'll be able to precipitate the DNA. So that's inorganic DNA isolation. And then the solid phase DNA isolation, so you have a spin column that has either the silica matrix, glass particle, and diatomaceous earth, or the anion exchange carrier. Then you'll be able to bind the DNA when you run the cell extract through the column, and later on you can elute the DNA. So this is just the procedure for the spin column to isolate the DNA. And this is to show you the anion exchange extraction, because DNA is negative charge, so that's why you use the anion exchange extraction, okay? Those are positive charge ion here can attract the DNA, and later on use a salt to elute the DNA. And then the precipitation. So basically, you have the salt, and then 100% ethanol, and then minus 20 degree, then you will be able to precipitate the DNA. All right. And then for the biotechnology technique, and you will need to use a different kind of the enzyme. So here is a list of the DNA manipulating enzyme that commonly used in the biotechnology. All right. So the nucleus. So nucleus basically degrade the DNA molecule by breaking the phosphodiester bond, either from the end of the DNA molecule, which will be the exonuclease, or to break the phosphodiester bond internally, that will be the endonuclease. Okay, and this is just the example for exonuclease to break the nucleotide one at a time from the end of the DNA molecule. So exonuclease only specific for the 3' end. Bio31 is used for both 5', 3', all right, to remove the DNA nucleotide. And endonuclease, so we have this S1 nucleus that can cleave this as single-stranded DNA. DNS1 cleave both single-stranded and double-stranded. And also we have restriction endonuclease, and restriction endonuclease, which is very specific to the depend on the type of restriction enzyme. They can recognize the target sequence and cut the specific sequence. Okay, the restriction enzyme basically discovered from the bacteria, because the bacteria protect land cell from the viral invasion. So that's why they have they develop this defense system to degrade the viral DNA. All right, so that's why we have restriction enzyme. And the type 2 is the most important one, because the enzyme recognition site and the cleavage site will be the same. And the 1 and 3 will be different, because 1, the recognition site, and the cleavage site will be 1,000 base pair apart, and 3 will be a 24 to 26 base pair apart. And this is just an example to show the type 2 restriction endonuclease. So basically the recognition site and the cleavage site will be exactly the same. So like a LU1 recognize AGCT, and then it's a symmetrical, also AGCT palindromic sequence, and then they will cut in the middle. And the EcoR1, they recognize the GAA-TTC, and then if you read backward here, GAA-TTC as well. And then they will cleave the DNA, all right, between the first one and second recognition, second nucleotide in the recognition site. And this is just detailed, like for example, the recognition site here, AAG, CTT is hexanucleotides, and most of the enzyme recognize the hexanucleotide, okay? And then they cleave this DNA, and then so you produce 2 DNA molecule here, and because this one, the single-stranded, we call this 5' overhand, and this one will recognize this sequence and cut here. When you dissociate, then you have this single-stranded DNA region, and these are the 3' overhand, 3' overhand. Or you can cut in the middle, then I give you a blunt end. Okay. The restriction enzyme activity that defined as 1 microgram of DNA in a total reaction volume of 50 microliter can be digested by the amount of enzyme in 1 hour. That will be 1 unit of the enzyme. And we'll skip this. So the methylation of DNA, so basically the methylation of DNA can protect the DNA from cleavage by its own restriction endonuclease. So that's the reason the bacteria cell, they develop the restriction enzyme. They will recognize the viral DNA from, distinguish the viral DNA from their own DNA. That's because their own DNA are methylated. so that they know that the messenger DNA is their own DNA. It's not a foreign DNA, all right? So they will not digest it. So that's how they recognize the host and distinguish the host from those foreign DNA. And the ligase, right? The DNA ligase, basically, they can repair the phosphodiester bond. Okay, so as you can see, let's say we have this the DNA that's been digested by the enzyme, and then you have two DNA molecule, and then you can replace the one of these DNA as long as they have the same complementary sequence, and then they can form this the hydrogen bond, and they stabilize these two molecules, and then in the meantime, DNA ligase can repair, fix the phosphodiester bond, sugar phosphate bond, all right, and then you have the complete DNA. Okay, so that's DNA ligase. For the cloning, so recombinant DNA, then you need to have sticky ends because that can increase the efficiency to ligate the two DNA molecule. So you have plasmid DNA and cloning. So the ligase here to repair the phosphodiester bond, all right? Skip this. And the production of recombinant DNA, so basically you have the vector which is the vehicle that can carry the foreign DNA, and then you can transfer the DNA into the any organism, right? So you cleave the DNA with a specific enzyme, and then the foreign DNA also has been treated by adding the same enzyme, so that they have the same cutting site, so that they can ligate this foreign DNA into the vector, right? In the presence of ligase. Okay. So the cloning of a cell, basically the cloning of cell, each individual cell, right? They divide many times so they can produce a colony of the progeny. And each colony is a clone, right? So this is cloning of cell. So plasmid. The plasmid, which is the vehicle that you can carry the foreign DNA and then introduce into the microorganism. And then the plasmid DNA must have some important feature so that you can use as the important tool to bring the DNA into the cell. So for example, the plasmid DNA need to have this gene for antibiotic resistance. This is a selective marker so that you can recognize which cell is recombinant cell, right? Through the antibiotic resistance. It's just, and then you, through the cloning, you put a foreign DNA here, and then perform the transformation into the bacteria cell. Okay. So the plasmid is a small circular piece of DNA, all right? And then you need to have these, original replication, antibiotic resistant gene, all right? And these multiple cloning sites, okay? So this is the overview of the plasmid that you can use to clone a specific DNA and then bring to the cell for, right, transformation. Multiple cloning site, I mean those are the restriction enzyme site, all right, polylinker, all right, polylinker or multiple cloning site are these group of the restriction enzyme. So you can design and to put the DNA into a lead either one of these locations, right? Perform the cloning. And this is an example of a pocket 19 vector, okay? And the PAC series of plasmid is very popular. They have extensive multiple cloning site. And this example to show you that they have, so although they have many cutting sites, they choose the Hindi 3, right? Hindi 3, and also the target DNA also cut with the Hindi 3. Right, so they need to be cut by the same enzyme so you can ligate this fragment into the vector. And so what you can use the restriction, you can use the to screen the presence of the recombinant DNA. So basically, you grow the cell in the antibiotic containing media, so that you can check the recombinant cell phenotype through the identification of colony on the selected media. Okay, and blue-white screen, basically, it's a basis for a selection to select the recombinant cell from the population that you use for the gene transformation. And for this experiment, so let's say we have this gene for the LEGZ, beta-galactosidase production, and ampicillin resistance. And then if you clone a piece of DNA into this location, right, into this location, then you break the LEGZ product, so that after you clone this, then you mix. Remember, when you mix the DNA with this vector, so some of the DNA will ligate this foreign DNA perfectly. Some of these will remain intact. Okay, they can remain intact, and then you have this mixture of the DNA, and you introduce this DNA into the cell. Then there will be a after transformation, there will be a 3 different possibility. So like here, no plasmid in the cell, or number 2, and the plasmid is inside the cell, but it's this format. Number 3 will be the plasmid inside the cell, but it is this format. So you can have 3 different possibilities. And then, so you can perform the blue-white screen, because this DNA is intact. They can produce LEGZ protein, beta-galactosidase, and this one will not. But this one carry the target sequence. So this one is intact, so they produce the beta-galactosidase. Then this enzyme will be able to cleave the F-scale chemical, so that they can produce the blue color. All right, so that's why you see this vector here. They produce the blue color. You can put the selected media with the amino ampicillin and also the F-scale, so that you can do this one-step screening. All right, so it's blue color that indicates the cell is a recombinant cell, but the DNA the cell carry is the original DNA. But for this one, you see the white colony, and this indicates that the cell is a recombinant cell, and the recombinant cell, they have the disrupted LEGZ, so that's why the colony is white here. But this plasmid has the ampicillin resistant gene, so they will be able to survive in the presence of ampicillin. Okay, and so the protein expression vector, so the plasmid vector, so like a PBR322, and the PAC, they are cloning vector. If you want to produce the product, the produced protein from the foreign DNA, then you will need the expression vector so that you can produce the protein. Okay, so it's just like this. You have the expression vector, and they can express the protein. So for the expression vector, okay, you need to have original replication, all right, and then multiple cloning site, at least one selectable marker, and also must be able to be transcribed in the host. That's the expression vector. And DNA library. Okay. And the PCR reaction. So PCR reaction. So it's an in vitro enzymatic reaction to amplify the specific DNA region. Okay, so that's a PCR reaction. So basically, PCR reaction require the oligonucleotide primer that is complementary to the template DNA, and so you can see the primer, so they will be able to synthesize a new DNA strand, right? And then the PCR reaction include 3 steps. So you have the denaturation, which means you need to denature this template DNA, and step 2, annealing the primer anneal to the template. Step 3 will be the extension. Extension, I mean the polymerization. So the primer can synthesize the DNA. Primer can synthesize DNA. Step 3. So you repeat this step, all right, for 20 to 50 cycles. Then you can produce millions copies of the specific DNA, your target DNA. Okay, so originally you have 1 double helix, all right, and then after 1 cycle, 2 to the power of 1, then you have 2 to the power 3 cycle, 4, all right, and 8. So that's the PCR, and then the quantitative PCR. Basically, you can monitor the DNA amount at the real time during the PCR reaction. So that's called a quantitative PCR. Okay, and quantitative PCR require the dye or the specific labeled probe so that you can evaluate the amount of the PCR, the amount of specific DNA produced by the PCR. Okay. And for the real-time PCR, it's a qPCR, because you need to monitor the amount of DNA at real time. So you need to have a dye so that you will be able to evaluate, because you can use ethylene bromide, high cybergreen, because this cybergreen, for example, all right, you add this cybergreen will bind to the double-strand DNA non-specifically. So during the denaturation, all right, so they release the cybergreen, and then annealing, so the primer anneal to the template start, and then synthesizing the new DNA, and then when you finish, then you have 2 double-stranded DNA. So the signal of the cybergreen will be double, all right? So that's the cybergreen to evaluate the amount of DNA. And for the qPCR, basically how you monitor the DNA at real time. That's because these dye that you added to the system, you can be recorded by the machine. All right, so basically it's depend on the threshold level. The threshold level, that means after you remove the background, right? Then that'll be the threshold level that's starting the amplification. And so here will be, let's say the threshold is here. Then that will be the first 0 cycle, and then 1, 2, 3 cycle. And then for this curve that show you the real-time DNA quantity, all right? And by looking at these difference, all right, the difference, the threshold between this 9 and this 9, the difference would be the number here, let's say, for example, 13, 11, so the number of cycle, the difference in number of cycle is 2 cycles, all right? And then that would suggest the initial amount of this 9 here would have 2 to the power of 2 times more material than the green one, all right? So that's how you evaluate the PCR result. And then, of course, you can use a TechMan, which is more specific, and they involve the extra probe. So as soon as you synthesize a new DNA strain, they will degrade the probe and release the reporter dye, the signal, so that you can detect. And the DNA fingerprinting. So basically, DNA fingerprinting, you digest the DNA with the enzyme, restriction enzyme, and then perform the gel electrophoresis. And after that, you transfer the DNA to a membrane, nitrocellulose membrane, and then you use the specific probe and to hybridize fluorescence labeled probe to hybridize the so basically after the gel electrophoresis, all right, and then you transfer the DNA to the membrane, and then the membrane, then you have the you can use the fluorescence labeled probe or isotope labeled probe to hybridize the target DNA sequence, all right, and then you'll be able to visualize the location of the specific DNA, and this is called a solid blotting. It's kind of a DNA fingerprinting. So this is the procedure. So you purify the DNA, perform the enzyme digestion, gel electrophoresis, transfer the DNA from the agar gel to the membrane, and then the membrane you will hybridize with the probe, a specific probe labeled with the fluorescence or the radioactive labeled isotope, and then you can visualize because the primer will find the complementary sequence on the gel, and then you'll be able to see the signal so that you can determine the location and the amount of that specific DNA in the sample. That's the fingerprinting, the solid blotting, all right? And then in organism with 2 sets of chromosomes, all right? And the given gene on one chromosome may differ slightly from the corresponding gene on the paired chromosome. And so these are known as the allele. So it can be homozygous, which mean those two are the same. Heterozygous, 2 are different. All right. And the restriction fragment of different size are obtained by treatment with endonuclease, and then to see the difference, that will be the RFLP, restriction fragment length polymorphism. So just like this, you have two copies, okay, 1 chromosome, 2 chromosome, and if you have mutation, then the enzyme will not be able to cut. If you have the wild type, then the enzyme will cut. All right, and then you run the gel electrophoresis, and then you see 2 band, and these 2 different number, and 1 band at the sum of these 2 fragments, and then this called RFLP, so that you can determine the identity. All right. So that's the RFLP, the sequencing. All right. So the sequencing, basically, you use the single-stranded DNA fragment as a template, and then to synthesize the new DNA strand. And in the meantime, you incorporate a specific nucleotide, and it's called a dideoxynucleotide. All right. And then by looking at different lengths of the the fragment, then you can determine the DNA sequence. And this is so-called a Sanger sequencing. Sanger sequencing because it's the dideoxy DNA nucleotide is used, and so for example, all right. So for example, the template, all right. So you have template, and then you start performing the synthesis, and you need to perform the 4 different reaction, all right. 4 different reaction. The first reaction, you add the 4 deoxynucleotide plus 1 specific dideoxynucleotide. So let's say it's a dideoxy A, all right. Dideoxy A. So that means the not only the 2' um, 2' carbon does not have the oxygen. Also, the 3' carbons hydroxy group has been replaced. Okay, the oxygen has been removed, and so that the dideoxy A, as soon as you incorporate the dideoxy A here, right, then the reaction will stop. No more DNA synthesis, and then after that you perform the electrophoresis. Then you can see the fragment, and then you can determine the size of fragment, and from the size of fragment, then you can interpret the sequence at that particular position, and that every fragment you see in the presence of dideoxy ATP, that will indicate that nucleotide is A, all right. And then if you have dideoxy GTP, then that'll be G and C, T, and so you can read the sequence from the bottom, all right? So you can read A, C, C, and then A, and then G, C, T, G, C, T. That's how you read the DNA sequencing, okay? So this is Sanger sequencing. And the proteomics is a study of the interaction among the all protein in the cell. That's called proteomic. Okay, the proteomic. And this is just the example of the Sanger sequencing. This in the old days, so that basically you have dideoxy A, dideoxy C, dideoxy G, and T. Each of them is labeled with the isotope label, so that you can perform the Sanger sequencing. So you can see all these fragments. That's different lengths of the fragment that has been incorporated to the last nucleotide is A, C, G, or T, so that you can read this sequence. And later on, they incorporate the fluorescence so that you can read the sequence through this capillary gel, all right? And so you can read the sequence, but the same principle, the Sanger sequencing, okay? And later on, the technique improved. They developed this parallel sequencing. Parallel sequencing. Remember, when you incorporate during the DNA synthesis, the hydroxy group, all right, will attack this alpha phosphate, release the pyrophosphate, all right, and then this pyrophosphate can be used, all right, in the presence of the ATP sulfurase, right, and also they can incorporate this pyrophosphate into this adenosine 5' phosphosulfate into the ATP, and this ATP can stimulate the luciferin, right, to react the reaction in the presence of luciferase, and then convert this luciferin into the oxyluciferin, and then release the light. So that every time if you incorporate a new nucleotide, then they will release the pyrophosphate, and then you will detect the light, so that you know the certain nucleotide has been added to the sequence. All right. So if let's say if you add DATP and then no signal, then you degrade this DATP, and then you add the second one, no signal, then you degrade this one. If you add the number 3, GTP, and then you see the signal, so that you know the next sequence is G, and then you continue the cycle. So ATGC again, if A show the signal, then you know it's A. So that's a parallel sequencing. And then after that, the past 10 years, so the technology improved dramatically. Then the the field developed this next generation sequencing, so NGS. So basically, there are different type of NGS technique. Can be the massively parallel sequencing. So that's the basically that be the advanced technique from the parallel sequencing, right? So they can perform like the hundreds parallel sequencing at the same time in a well. So that's why it's called a massively parallel sequencing. Or you can do the sequencing by synthesis. So that would be similar to the Sanger sequencing, and instead of the dideoxy nucleotide they use the regular deoxynucleotide, but they engineer to have the block in the 3' hydroxy group, and also they have a specific fluorescence label to the nitrogenous base. So as long as you incorporate a specific nucleotide, they detect the signal, right? And then after they record the signal, they remove the block from the 3' the 3' carbon, and then they can continue to add the next sequence. So that's a sequencing by synthesis. And then also you can have sequencing by ligation, and this will be special because this one does not require the DNA polymerase. They use a ligase to perform the sequencing, and the sequencing by semiconductor. So basically, if you remember this particular reaction here, so if you add the DNA and the incoming nucleotide, so the hydroxy group attack the alpha phosphate, release the pyrophosphate. In the meantime, this hydroxy group here, the hydrogen will be released. All right, the hydrogen will be released, so that will cause the pH increase. Okay, and then you can detect the change of pH, so that you know which nucleotide is added to the sequence. All right. So that's basically the NGS sequencing, and the microarray basically is like the massive amount of the southern blot analysis, because you can have the slide, and then the slide you have like thousands of primer, a probe on the slide, and then you have the sample, and then hybridize with the probe. And then later on, of course, those are sample are labeled with the fluorescence. Okay, either Sci5, Sci3 fluorescence, and then you can determine the amount of the DNA bind to the particular probe, so that you know either the copy number of the particular DNA, or the expression level of the particular gene. So that's a microarray. Okay, so that'll be the end of the chapter 13. Okay, any question? Adele.